Welcome to AP Biology, topic 2.1. My name is Paulette Unger, and I teach at Christ Church Episcopal School in Greenville, South Carolina. In this lesson, I'm going to discuss how ribosomes reflect the common ancestry of all known life, and then I'll discuss the structure and function of a few other subcellular components and organelles. All living cells contain a genome and ribosomes, reflecting the common ancestry of all known life. So we have prokaryotic cell in figure 1 and a eukaryotic cell in figure 2. And the presence of these two components in all known life makes sense because all cells have to have mechanisms to store and pass along their genetic information and nucleic acids, and ribosomes work hand in hand to get this job done. Ribosomes function to synthesize proteins according to messenger RNA sequences, and the instructions that are encoded in that messenger R sequence originate from the genome of a cell. Now, unlike prokaryotic cells, the genome of eukaryotic cells is enclosed in a membrane-bound structure called the nucleus, which you can see here in the figure 2, which is a distinguishing feature of eukaryotic cells. However, the ribosomes, which are found in both cell types, are not membrane-enclosed structures. So let's take a look and see what the structure of ribosomes is actually like. So ribosomes consist of two subunits, and you can see here in this image, it has a small subunit and a large subunit that come together. And like I mentioned, they're not membrane-enclosed subunits. And ribosomes are made of ribosomal RNA, or rRNA, and proteins. And as I mentioned in the previous slide, the ribosomes are going to make proteins for the cell according to the messenger RNA sequences. So you can see a polypeptide chain growing from the ribosome. Now, eukaryotic cells have additional subcellular structures that are membrane-enclosed organelles, and each of those performs specialized functions for the cell, and these compartments increase the efficiency of the cell's metabolic reactions. So I hope you're ready because I'm going to go through a whole bunch of these for you right now and highlight the structure and function. So here we have the endoplasmic reticulum, or the ER, which is a network of membrane tubes within the cytoplasm of eukaryotic cells. And there's two forms of the ER. So you can see here we have the rough ER, which in the image, notice it's like studded with little dots. Well, those dots represent ribosomes, which are attached to the membrane of the rough ER. And the rough ER compartmentalizes the cell and is associated with the packaging of the newly synthesized proteins made by those attached ribosomes for possible shipment to other parts of the cell, or even for export out of the cell through the plasma membrane. The other form of the cell is the smooth ER, and if you look at the picture, notice it doesn't have those little dots on it, and so the smooth ER does not have ribosomes attached to it, but functions include detoxification for the cell, so like detoxifying waste or drugs, and it also functions in making lipids for the cell. So notice again, we have that structure function theme, like structural differences between that rough ER and the smooth ER leads to their functional differences. Next, we have here the Golgi complex, which is a series of flattened membrane sacs found in eukaryotic cells. And the Golgi is involved in the correct folding and modification of newly made proteins and packaging those proteins for protein trafficking. Again, shipping proteins to other parts of the cell or even for export out of the cell. So you'll notice these little bubbles around the Golgi complex. These are vesicles, and you have incoming transport vesicles and secretory vesicles. And vesicles are just membrane containers that help move materials from one part of the cell to the next. So I decided here to have a little fun with some Play-Doh and create a short stop motion video clip for you to let you see how the Golgi works in action. So it helps me a lot of times to see things in motion. So while you're watching this, you have a vesicle containing a protein. Look closely through the animation as the vesicle fuses with the Golgi membranes, and that protein bead like moves through the different membranes and the modification occurs. And you'll have a secretory vesicle coming out the other side, shipping it to the plasma membrane. Okay, wait for it. There's your modification. And now you have that secretory vesicle. Okay, so that just gives you an idea of how proteins might move through the Golgi membranes and get modified. The next membrane enclosed organelle we have is the mitochondria, and mitochondria actually have double membranes. So we have an outer membrane and an inner membrane. The outer membrane is smooth, while the inner membrane is highly convoluted, forming folds called cristae. And the mitochondria functions in production of ATP energy for the cell that the cell can then use to do work for itself. Here we have the lysosome, which is a membrane-enclosed sac found in some eukaryotic cells that contain hydrolytic enzymes. So hydrolysis is chemical digestion. And so what might the cell be digesting? Well, a variety of materials such as damaged cell parts or macromolecules such as carbohydrates, or proteins, lipids. So the lysosomes function in that regard. Vacuoles are membrane-bound sacs found in eukaryotic cells, and they play a variety of roles, ranging from the storage of water and other macromolecules to the release of waste from the cell. And in figure 1, we have a plant cell, and plant cells have large central vacuoles. And I thought this was a really cool image of plant cells from the microscopic view, and you can see nicely the membrane of the vacuole containing that fluid for that plant cell. Lastly, we have chloroplasts, which are found in photosynthetic algae and plants. They have a double outer membrane, much like the mitochondria does, and they're specialized for capturing energy from the sun and producing sugar for the organism through the process of photosynthesis. So we just looked at a bunch of visual representations of several membrane-bound organelles and subcellular parts. So let's practice using the visual representations and describing biological concepts from those visuals. Here we have an AP question. I'm going to ask you to pause the video, read the question, take a look at the picture of the cell, read the labels carefully, and select your best choice for an answer, and then press play, and we'll discuss it together. Okay, so let's see what you have. If you chose B, you are correct. So it says, which of the following best describes how these three structures indicated by arrows work together? Well, we have the rough ER, and the rough ER has ribosomes embedded in its membrane. So proteins are synthesized there, and the rough ER can ship those proteins in a vesicle to the Golgi, and the Golgi can modify those proteins, package them again in a vesicle for secretion from the cell or use from the cell. So choice B summarizes the relationship of those three best. In this case, to synthesize and isolate proteins for secretion or use for the cell. So you just keep in mind, like, think if you, as you're learning the functions of these, that'll help you determine the best way that they might work together. So some key takeaways here are that ribosomes are not enclosed by a membrane and are subcellular components found in all forms of life, which reflects that common ancestry of all known life, and they function to make proteins for the cell. Eukaryotic cells have additional membrane-enclosed organelles that perform specialized functions for the cell as well, and we went through several of them that are listed here. So feel free to go back at any point in time and review those structures and functions as you need. Thank you for watching, and I hope this video was helpful to you. Welcome to AP Biology, Cell Structure and Function. My name is Paula Dunger, and I teach at Christ Church Episcopal School in Greenville, South Carolina. In this video, I'm going to discuss the subcellular components and organelles and their function and how they interact to contribute to the overall function of the cell. Then I'll also highlight the structural features of a cell that allow organisms to capture, store, and use energy. And so let's actually start with that. What are these energy capturing organelles? So first we have the chloroplasts, and chloroplasts are specialized for photosynthesis and capturing energy from the sun to produce sugar. Within the chloroplast are two distinct compartments. First, we have the thylakoid, as you see here in the image to the right, and these are highly folded membrane compartments that are organized into stacks called grana. Membranes contain chlorophyll pigments that comprise the photosystems, and electron transport proteins can be found between the photosystems embedded in these thylakoid membranes. This is where the light-dependent reactions of photosynthesis occur, and the folding of these internal membranes increases the surface area and efficiency of these reactions. There's also within the chloroplast the stroma, and this is the fluid between the inner chloroplast membrane and outside the thylakoids. And in the stroma is where the carbon fixation reactions occur. You can see that here on the diagram. Now, the mitochondria is the other energy capturing organelle, and the mitochondria have double membranes, which provide compartments for different metabolic reactions. So we have the outer and inner membrane shown here, and the mitochondria capture energy from macromolecules such as carbohydrates, but can also get energy from proteins or lipids as well. The Krebs cycle reactions occur in the matrix of the mitochondria, the fluid-filled portion that you see labeled in our diagram, while the electron transport and ATP synthesis occurs in the inner mitochondrial membrane. And this membrane is also highly folded, which increases the surface area to allow, in this case, more ATP to be made. And so you can see those folds there labeled cristae. Now, additional organelles are found in eukaryotic cells. So let's look at the structure and function of the vacuoles. Vacuoles play a variety of roles, including storage and release of water, macromolecules, and even cellular waste. So here we have this sad-looking plant. So let's see why this plant is wilted. What might be the role of a vacuole in this scenario? Well, in plants, vacuoles aid in retention of water for turgor pressure. And turgor pressure is an internal cellular force that's usually caused by water pushing up against the plasma membrane and the cell wall. So if we look at a microscopic image of plant cells, here we have, let's say, cell A. You can see the membrane of the vacuole, and notice it's not pushing up against its plasma membrane and cell wall. However, here in cell B, notice that you can't see clearly the membrane of the vacuole because the vacuole is more filled with fluid and pushing up against its membrane and cell wall. So therefore, cell B would have more turgor pressure, and if more and more cells look like cell B in this plant, it might not look so wilted like we see here. Next, we have the lysosomes. Lysosomes contain hydrolytic enzymes, and these enzymes can be used for chemical digestion. So what might be some things that the lysosome is digesting for the cell? We've got macromolecules, other worn out cell parts and organelles. So you can recycle these organic materials. And even the lysosome could lead to programmed cell death or apoptosis. Next, we have the ER, which performs several functions for the cell, including mechanical support. So this extensive network of membranes can provide mechanical support for the cell. It can play a role in intracellular transport and shipping materials to various parts of the cell. And the rough ER, which you see here, has ribosomes attached to it, and so it can carry out protein synthesis on those ribosomes. So we just talked about several organelles and their structure and function, but I want to highlight for you how these cell parts can actually interact together to help the cell maximize its efficiency and perform a diverse array of functions. So here I made a stop motion video for you, so you might want to grab some popcorn. I'm not sure it's popcorn worthy, but we'll see. So here we have the rough ER in this video, and the dots represent the ribosomes, and there's a protein in there that was made from those ribosomes. We have the Golgi and the plasma membrane. So as you watch, take notice of how the protein is shipped using membrane-enclosed vesicles from the rough ER to the Golgi, and then from the Golgi, where it gets modified, to the plasma membrane. So there you have it. So you have a lot of interactions between these different cell parts through the use of vesicles. And I was having so much fun that I made another video example for you. So in this video, we're going to look at a lysosome and its interaction with a vesicle that contains a macromolecule. This macromolecule could have been ingested by the cell, or it could have come from another internal cellular part. So watch as that vesicle fuses with the lysosome, and the lysosome that contains hydrolytic enzymes does its thing and digests it. So there the hydrolytic enzyme is digesting it, and now you have these organic monomers. And these organic monomers can even be released by the lysosome and be used by the mitochondria as a source of energy for the cell. Now let's practice the skill of argumentation. When initially constructing an argument, it's important to make a claim. And a scientific claim is just a statement based on evidence. So here is an AP question that presents you with some data that you can use as evidence to make a claim and draw a conclusion. So I'm going to ask you to pause the video right now, read through the question, look at the data, select your best choice, and press play, and we'll talk about the answer together. Okay, so hopefully you have answer choice ready. And the correct answer here is B. So which of the following conclusions about the radioactively labeled amino acids is best supported? Choice B, in which it was mostly incorporated into proteins that regulate and manage metabolic activities. So why is that? Well, amino acids are the building blocks of proteins, and the data indicate that most of the radioactively labeled amino acids were in the mitochondria, with 2.7 in terms of a relative amount compared to the other. So therefore, the data best supports the conclusion that most of the amino acids and molecules were incorporated into proteins that regulate and manage metabolic reactions, since the mitochondria is involved in producing energy for the cell. So some key takeaways here are the subcellular components and organelles interact to support cell function. We went through several of those structures and their function and how it helps increase the efficiency for the cell to perform chemical reactions and store materials. Specifically, we looked at how the chloroplast and mitochondria are structural features of eukaryotic cells that allow organisms to capture, store, and use energy. And the folding of the inner membrane in both of those structures increases the surface area, which will allow more ATP to be synthesized for the cell. Thank you for watching, and I hope this video was helpful. Welcome to AP Biology. My name is Paula Dunger, and I teach at Christ Church Episcopal School in Greenville, South Carolina. Have you ever wondered why cells are typically small and why we don't see giant unicellular organisms roaming the planet? Well, we're going to explore why that is in this lesson and the role that the surface area to volume ratio of cells plays in that cell size, as well as how that ratio is determined. So cells are typically small because moving materials such as nutrients and waste in and out of cells gets more challenging the larger the cell is. And so I want to walk you through a mini demonstration that I performed at home to illustrate how the size of the cell can impact its efficiency in transporting materials across its surface. So in figure 1, you see 3 various sized cubes that I cut from beets, which are root vegetables, as shown in figure 1. And these cubes are going to represent 3 hypothetical cube-shaped cells with sides 0.5 centimeter, 1 centimeter, and 2 centimeters in length. And I left these cubes in this dish, and then I poured bleach over them to cover them completely, and I let them sit for 30 minutes to soak. And I used bleach because the beet cubes will turn white when exposed to the bleach, so it would be easier for me to observe how much of the bleach was able to move throughout each cube. So you ready to see the results? Let's check it out. So since there was a higher concentration of bleach outside the cubes, bleach was able to move across the surface of each cube, as you can see here by the white coloration of each cube. But I was curious how much bleach actually was transported throughout the entire cube. So I cut each cube in half, and this is what I saw. So each cube had a different amount of bleach after the 30 minutes, and the smallest cube had 96% of its total volume containing the bleach, compared to only 39% of the larger cube containing the bleach. So the smaller cube was more efficient at allowing the bleach to enter, as shown by that higher percentage of its total volume that contained the bleach. So cells need a sufficient amount of surface for exchanging materials with its surrounding environment. And we can quantify the amount of surface available for materials to move by calculating the surface area of the cubes in this case. The surface area of a cell is defined as the amount of surface covering the outer part of the cell. But it's not only about the surface area. We also have to consider the volume of the cube, or the amount of space that the entire cube or hypothetical cell takes up. By looking at the relationship between surface area to volume, we can get a better indication of the efficiency of that cell at moving materials across its surface. The larger the ratio, the more efficient the cell would be. So the formulas for surface area and volume of a cube, as well as for other shapes like cylinders and spheres, can be found on the AP formula sheet on AP Central. And I have three cubes that are not drawn to scale, but they're going to represent our beat cubes that were used in the demonstration. So I'm going to calculate the surface area and volume for the smallest cube to show you how to apply these equations. So the surface area of a cube is 6 sides squared. We have sides squared of one face, and there's 6 faces to the cube. So we're going to multiply our side squared by 6. So we have 6 times 0.5 centimeters squared. So 0.5 times 0.5 is 0.25 centimeters squared, and we'll multiply that by 6 to get a surface area of 1.5 centimeters squared for our small cube. And I filled that in the table for us. The volume of the cube is side cubed, so we have 0.5 times 0.5 times 0.5 will give us 0.125 centimeters cubed. And now to set up the ratio of surface area to volume, we're going to put 1.5, the surface area, in the numerator, and then we'll put the volume of 0.125 in the denominator, and that gives us a surface area to volume ratio of 12:1 for our smallest cube. So I'm going to ask you to pause the video now, calculate the surface area and the volume and the ratio for the 1 centimeter and the 2 centimeter cube so we can see how size influences this ratio as a cell gets larger. When you have your answers, just press play and we'll check your work. Okay, so let's see what you have. For this 1 centimeter cube, the surface area is 6 centimeters squared, and the volume would be 1 centimeter cubed. So when we set up our ratio of surface area to volume, we get 6:1 as our ratio for our 1 centimeter cube. For our 2 centimeter cube, should have gotten surface area of 24 centimeters squared and a volume of 8 centimeters cubed. So when we set up our surface area to volume ratio, we get 24:8 or 3:1. So if we look now at the trend, as the cells got larger, as our cubes got larger, the surface area to volume ratio decreased. Our smallest cell had a ratio of 12:1 compared to the largest cell with a ratio of only 3:1. So smaller cells typically have higher surface area to volume ratios and therefore are more efficient at exchanging materials with the environment. And so if you think of our cube, if our smallest cube was the real cell, it would have been able to supply its internal demands of taking in oxygen and nutrients if this was a real cell, as well as removing waste and CO2 and heat. The smaller cell would have done a lot better for itself compared to our larger cell. Because as the cell increases in volume, the relative amount of surface area decreases, making it difficult for larger cells to meet those demands. And so these limitations are going to restrict cell size and shape. And I will share with you some examples of modifications that cells and organisms have to overcome this challenge in the second lesson for this topic. Scientists can use visual representations to represent relationships within biological models, including mathematical models. So let's take a look at an AP question. Below is a diagram of a cell increasing in size. Which of the following graphs best represents the effect of the cell's increasing size on its surface area to volume ratio? So I'm going to ask you now to pause the video and select your best choice and then press play and we'll see what we have. Okay, so the best representation of this relationship is D. As the length of the side of a cube-shaped cell increases, the surface area to volume ratio decreases. Some key takeaways from this lesson are smaller cells typically have a higher surface area to volume ratio and are more efficient at exchanging materials with the environment. The surface area to volume ratio can affect the ability of a cell or organism to obtain necessary nutrients, eliminate waste, and acquire and dissipate thermal energy. We also practice using formulas that can be found on the AP formula sheet for surface area and volume of a cube. And then we use that calculated surface area to volume to determine the ratio. Thank you for watching, and I hope this video is helpful. And if you need more time, just rewind. Welcome to AP Biology, Cell Size. This is video lesson 2, and my name is Paulette Unger from Greenville, South Carolina. In this lesson, I'm going to share with you some examples of structural modifications of cells that increase their surface area, as well as specialized strategies used by cells and organisms for more efficient exchange of molecules with the environment. I'll also discuss with you the surface area to volume ratio and how it affects the rate of heat exchange with the environment for organisms. So the surface area of the plasma membrane must be large enough to efficiently exchange materials. Smaller cells typically have a higher surface area to volume ratio and are better able to do this. As cells increase in volume, the relative surface area decreases, but the demand for internal resources like taking in oxygen and nutrients, as well as dissipating heat and ridding itself of waste and carbon dioxide, that demand for those resources increases as the cell gets larger. So more complex structures are necessary to adequately exchange materials with the environment. So let's look at an example in plants to start. Membrane folding can increase surface area. And if we look here at figure 1, we see the roots of a plant and they look all a little fuzzy, right? Well, these are root hairs that are on the surface of plant roots. And these root hairs increase the surface area of the root, which can allow for increased absorption of water and nutrients. And if we look at the root hair cell in figure 2, you can see how that membrane is extended and that increases that surface area for water to enter. Another example of membrane folding can be found in the small intestines. After chemical digestion occurs in the digestive tract and macromolecules are broken down into monomers, those monomers need to be transported across the surface of the small intestines and into the bloodstream so they can get transported to cells, and cells can use those monomers as energy and do cell work. And so what we have here is a modification on the outer lining of the small intestines where the lining is highly folded into these finger-like projections called villi. But it doesn't just stop there. Each of the villi has additional microscopic projections called microvilli, as shown in figure 1, that further increases the surface area for the absorption of these nutrients. So this increase in folding, the more folds you have, the more efficient these cells would be at, in this case, transporting nutrients across their surface. And imagine if there was a condition where there was damage to these villi and the villi flattened, like as shown here in figure 2. Any loss of membrane folds like this would decrease the efficiency of the cell in absorbing nutrients for the organism. Also, as organisms increase in size, their surface area to volume ratio decreases, and this can affect properties like the rate of heat exchange with the environment. So let's take the elephant, for example. Okay, these are really large animals, and their body have little surface area relative to the enormous volume that they contain. So it's difficult for the elephant to release heat from their bodies across their surface. So they have an adaptation. They have their large flat ears that you see here, and that's going to allow the elephant to dissipate more thermal energy as blood flows closer to the surface. So they're able to overcome this limitation by having the larger, flatter ears. And then they'll end up cooling off for themselves a little bit more. Organisms also have evolved highly efficient strategies to obtain nutrients and eliminate waste. And so we can see that here we have our magnified leaf, and notice on the surface of the leaf we have these openings called stomata. And so these stomatal openings of the leaves help them obtain molecules from the environment as well as release molecules. So in our image, we see that when the stomata are open, CO2 can enter the leaf and oxygen and water are being released into the atmosphere. I do want to point out, though, that when stomata are open, this image just shows CO2 coming in, but oxygen gas can also come in. It's a two-way flow. So oxygen and CO2 can come in just as much as oxygen and CO2 can be released from these openings. So scientists are collecting data all the time, and then they have to analyze that data and they perform statistical tests. So performing mathematical calculations, including ratios, is a common practice. And so let's take a look at an AP question where you're going to be calculating the ratio. So I want to going to ask you to pause the video now, take a few moments, read the question, perform some calculations, select your best answer, and then press play when you're ready. Okay, so the question asks, which of the following best describes how the ratio of the density of stomata per CO2 concentration changes as the CO2 concentration increased? The answer is A. The ratio decreases from 0.86 to 0.63. So we're setting up that ratio of density of stomata, which would be in your numerator, per CO2 concentration, which would be in your denominator. So when the CO2 concentrations were less, you would have taken 300 stomata per millimeter squared and divided that by 350 parts per million CO2 to get 0.86. And then you would take 250 stomata per millimeter squared and divide that by 400 parts per million CO2 to get 0.63. So that's how those ratios would have been set up and calculated to get that answer, which is choice A. Okay, so some key takeaways from this lesson: as cell volume increases or a cell becomes specialized for transporting across its surface, like the small intestines or the roots of plants, structural modifications through all that membrane folding are necessary to help them adequately exchange these molecules. As organisms increase in size, their surface area to volume ratio decreases, making it harder for them to release heat. But adaptations, like we saw with the elephant and having the larger, flatter ears, may improve an organism's efficiency at exchanging that heat energy. And cells and organisms are going to use specialized exchange surfaces, such as stomatal openings on the surface of a leaf, in order to maximize exchanging molecules, taking in molecules, releasing them as needed. To help the organism function most efficiently. Thank you for watching, and if you need more time, just rewind.
Welcome to AP Daily. This is Mr. Burris from Union High School. Today's topic is plasma membrane. So let's dive in. Today's topic, what will we be learning? Well, we're going to look at the structure of phospholipids and how they establish and maintain the internal environment of the cell. We'll look at the roles that proteins play in maintaining the internal environment. And finally, we'll look at the structure and framework of the fluid mosaic model of the cell membranes. So the cell membrane provides a boundary between the interior of the cell and the outside environment. This allows the cell and the cell membrane to control the transport of materials in and out of the cell. So what are these cell membranes made up of? They're made up of phospholipids. Phospholipids are amphipathic. They're both hydrophilic and hydrophobic at the same time. So for these phospholipids, the hydrophilic phosphate head is polar and the hydrophobic fatty acid tail is nonpolar. These phospholipids spontaneously form a bilayer in an aqueous environment as shown on your screen. The tails are located on the inside of the bilayer, interacting with each other in a hydrophobic way. And then the heads are exposed to the aqueous outside and inside environments of the cell. In the cell membrane, there are embedded proteins. These embedded proteins can be hydrophilic or hydrophobic, and there's two types. The first type is peripheral proteins. These peripheral proteins are loosely bound to the surface of the membrane, and they're hydrophilic with charged and polar side groups that allow them to interact with the hydrophilic region of the phospholipid bilayer. The other type of protein is the integral protein. The integral proteins span the membrane, and they are hydrophilic with charged and polar side groups on the ends when they interact with the hydrophilic regions of the phospholipid bilayer, but they're also hydrophobic with non-polar side groups that penetrate the hydrophobic interior of the bilayer. An example of an integral protein is the transmembrane proteins. These proteins have a variety of functions. We're going to just name 6 of them. The first function, transport. Another function would be cell-to-cell recognition. A third function, enzymatic activity. A fourth function, signal transduction. Another one would be intercellular joining. And finally, you have attachment for extracellular matrix or cytoskeleton. So what does the fluid mosaic model mean in terms of the cell membrane? It means that it is structured as a mosaic of protein molecules in a fluid bilayer of phospholipids. The structure is not static. It is held together primarily by hydrophobic interactions, which are weaker than covalent bonds. This allows most of the lipids and some of the proteins to shift and flow along the surface of the membrane or across the bilayer. So one of the parts of the fluid mosaic model is cholesterol. And what cholesterol does is it's a type of steroid that is randomly distributed along the phospholipid bilayer, and it's wedged between these phospholipids in the cell membrane of eukaryotic cells. And what it does is it helps regulate the bilayer fluidity under different environmental conditions. The last component of the fluid mosaic model would be carbohydrates. The diversity and location of these carbohydrates allow them to function as cell markers, and there's two types. Type number one, glycoproteins. These are carbohydrates attached to embedded proteins. The other one is glycolipids. These are where the carbohydrate is attached to a lipid in the bilayer. So with that being said, let's practice what we learned today. The skill we're going to focus on in this lesson today is visual representation. You're going to describe the characteristics of a biological concept, process, or model represented visually. So let's start. I'm going to have you pause your video, make sure you analyze the figure as you read the question, and then jot down your answers. When you are ready, push play again, and we'll go over the answer. Welcome back. In this question, you're looking to describe the model that best represents the structure of the plasma membrane in a non-aqueous environment with a cytosol environment similar to living cells, meaning aqueous. So the model that best represents that is E. Since the phospholipid heads are orientated towards the aqueous environment and the hydrophobic tails are orientated towards the non-aqueous environment. So what should you take away from today? Well, first, that the phospholipid spontaneously form a bilayer in an aqueous environment with the hydrophilic heads on the outside and the hydrophobic tails on the inside. The embedded proteins can be hydrophilic or hydrophobic. The embedded proteins also have a variety of functions ranging from transport to signal transduction. And finally, the fluid mosaic model consists of a structural framework of phospholipid molecules that are embedded with proteins, steroids such as cholesterol, glycoproteins, glycolipids that can flow and shift within that cell membrane. Well, thank you for your time. I hope it was worth your time. Until next time, thank you. Welcome to AP Daily. This is Mr. Burris, and we're going to be covering membrane permeability. So what will we be learning today? Today's lesson will cover the structure of the cell membrane, how it is involved in selective permeability, and how it separates the internal environment of the cell from the external environment, the fluid mosaic model, and what type of molecules can pass freely across the membrane, and what type of substances need some help with embedded channel and transport proteins. And finally, we'll look at cell walls and how they form boundaries. So let's begin. So as you recall, the structure of the cell membrane is made up of phospholipids, and these phospholipids are amphipathic. Well, what does that mean? Well, that means they're hydrophobic and hydrophilic at the same time. These phospholipids spontaneously form a bilayer in an aqueous solution, as shown on your screen. The fluid mosaic model is this moving phospholipid bilayer composed of molecules such as proteins, steroids, and carbohydrates. The selective permeability of the cell membrane is the direct consequence of this membrane structure. So what things get across the cell membrane? Well, small nonpolar molecules are able to pass freely across. What are examples of those? Nitrogen, oxygen, and carbon dioxide. So what happens if they can't get across the membrane? Well, hydrophilic substances such as large polar molecules and ions cannot freely move across the membrane. They need some help, and that help comes through transport proteins. There are two types of transport proteins. First one, a channel protein, which is a hydrophilic tunnel spanning the membrane that allows specific target molecules to pass through. The second one is a carrier protein, which spans the membrane as well and changes shape to move a target molecule from one side of the membrane to the other. Small polar molecules, like water, can pass directly through the membrane in minimal amounts, but it's really, really small. So what is the purpose of a cell wall in some cells? Well, it's a structural boundary and permeable barrier. As a structural boundary, as in the case of a plant cell, it protects and maintains the shape of the cell. It prevents against cellular rupture when internal water pressure is high. It helps the plant stand up against the force of gravity. Now, as a permeable barrier, things can't get through, but the way they get through is through these small holes called plasmodesmata, which are between plant cells that allows for the transfer of nutrients, waste, and ions. However, one thing I do want to point out is do remember that animal cells do not have these cell walls. The cell walls are composed of complex carbohydrates. You will find cell walls in plants, and they're made up of cellulose, which is a polysaccharide. You'll find them in fungi, and they're made up of chitin, which is another polysaccharide. And finally, you'll find them in prokaryotes in the form of peptidoglycan, which is actually a polymer consisting of sugar and amino acids. So let's practice what we learned today. Today's skill focus is on statistical test and data analysis. Use data to evaluate a hypothesis or prediction, including supporting or refuting the alternative hypothesis. So let's begin. What I would like you to do is you're going to pause your video, you will analyze the table, and you will read your question. Once you feel comfortable, select your answer and then push play again. Welcome back. For this question, you were looking at which of the following identifies the treatment group most likely to have provided an unreliable estimate of the mean and correctly explains why the estimate appears unreliable. So your answer should have been A, treatment group 2. The reason for that is the lower than expected mean absorption compared to the rest of the treatment groups and the higher standard error found in the table. So what should you be taking away from this lesson today? The structure of the cell membrane results in selective permeability. The selective permeability is a direct consequence of membrane structure. Small polar molecules, including nitrogen, oxygen, carbon dioxide, can freely pass across the membrane. Hydrophilic substances, however, such as large polar molecules and ions, need to move through embedded channel and transport proteins. Polar uncharged molecules, including water, can pass through the membrane, but in small amounts. The cell walls provide a structural boundary as well as a permeability barrier for some substances to the internal environments of the cell. The cell walls of plants and prokaryotes and fungi are composed of complex carbohydrates. Well, I hope this video and lesson was informative to you. Until next time, thank you. Welcome to AP Daily. This is Mr. Burris, and we're covering membrane transport. So today, what will we be learning? In today's lesson, we'll take a look at passive and active transport and how they move molecules across the membrane. We'll look at how concentration gradients are formed, and then finally, how large molecules move in and out of the cell. So let's dive in. A concentration gradient is when a solute is more concentrated in one area than another. A membrane separates the two different concentrations of molecules, allowing for that concentration gradient to form. So that leads us to passive transport. Passive transport is the net movement of molecules from a high concentration to a low concentration without metabolic energy, such as ATP, needed. This plays a primary role in the import of materials and the export of waste. There's two types of passive transport. The first type is diffusion, where the movement of molecules from high concentration to low concentration is just directly across the membrane. What type of molecules do that? That would be small nonpolar molecules such as nitrogen, oxygen, carbon dioxide. But what if their molecules are not small or nonpolar? Well, They then move through via a process called facilitated diffusion, which is, again, the movement of molecules from a high concentration to a low concentration, but it's done through transport proteins. It allows for hydrophilic molecules and ions to pass through the membrane. So now let's take a look at an example. In this example, we have a plastic box with a piece of plastic in the middle with small little pores in it. And on one side of that simulated membrane are small white molecules and then larger red molecules. So let's take a look, see how passive transport works. So you'll see in the video that the small white molecules were able to get through the membrane because they were small enough and they went from their high concentration to a low concentration. The larger red molecules are still in a high concentration on one side because they were not able to get through the membrane. Now, you might be thinking, how is that passive transport when we were shaking the box to get things to move through? Well, it's just demonstrating an idea called Brownian motion, which is the random, uncontrolled movement of particles in a fluid. So what happens if the cell wants to move molecules the other direction? Well, that's where active transport comes into play. Active transport requires the direct input of energy, such as ATP, to move molecules from regions of low concentration to high concentration. So they're moving against their concentration gradient. So talking about smaller molecules, what about large molecules, such as large macromolecules? What do the cells do with those? Well, in endocytosis, the cell uses energy to take in macromolecules and other matter by forming new vesicles derived from the plasma membrane. And there's three types. The first type is phagocytosis. It's where the cell takes in a large particle, which you can see on your screen. The next one is pinocytosis, where the cell takes in extracellular fluid containing dissolved substances, as you can see on the screen. Finally, you have receptor-mediated endocytosis. This is where a receptor protein on the cell is used to capture specific target molecules to bring into the cell through a vesicle. Well, that's bringing large molecules in. Well, what happens when you want to remove large molecules? Well, in exocytosis, internal vesicles use energy to fuse with the plasma membrane and secrete these large macromolecules out of the cell. Examples: proteins such as signaling proteins, hormones, and waste are examples of things that the cells would move out through exocytosis. With that being said, let's practice what we learned today. In today's lesson, the focus is questions and methods. You're going to propose a new or next investigation based on an evaluation of the design methods. So what I would like you to do on this question is hit the pause button, read through it, and come up with your answer. Welcome back. In this question, you were looking at what could the scientists propose as a next investigation to help determine whether the organism used endocytosis to eat the bacteria. After reading through the choices, you should have come up with B as the best answer, because the number of vesicles before and after would indicate an increase in endocytosis occurring, so the increase of eating more organisms and more bacteria. So what should you take away from today's lesson? Today's lesson included passive transport, which is the net movement of molecules from high concentration to low concentration without any energy. Active transport requires the direct input of energy to move molecules from regions of low concentration to high concentrations. The membrane allows for the formation of concentration gradients. In exocytosis, the cell removes large molecules through the plasma membrane. And in endocytosis, the cell uses energy to bring in these large macromolecules via the plasma membrane, forming vesicles. Well, I hope this video was informative to you. Until next time, thank you. Welcome to AP Daily. This is Mr. Burris, and today we're talking about facilitated diffusion. So, what will we be learning today? For today's video, we're going to look at why are membrane proteins required for diffusion of certain molecules? How do large quantities of water move across the membrane? How do membranes become polarized? When is metabolic energy, such as ATP, needed for transport? And how does the sodium-potassium pump contribute to membrane potential? So let's dive in. Membrane proteins are necessary for facilitated diffusion. Facilitated diffusion is this movement of molecules from a high concentration to a low concentration through transport proteins. Large and small polar molecules need channel or carrier proteins that get across the membrane. Water in large quantities pass through aquaporons. Charged ions, including sodium and potassium, require channel proteins. So membranes may become polarized by the movement of ions, which help in the formation of gradients. One type of gradient is an electrochemical gradient, which relies on membrane potential, which is an electrical potential difference across a membrane. Membranes may become polarized by the movement of ions across the membrane. As shown in the illustration, the sodium channels open up, they diffuse across, and the potassium flows the other way due to the membrane potential and concentration gradient established across the membrane. So let's look at an example. For the sodium-potassium pump, it contributes to the maintenance of the membrane potential. How does it do that? Well, it takes 3 sodiums and pumps them against their concentration gradient, as shown in the picture, to establish a concentration gradient of sodium. And then it also pumps 2 potassium ions the other direction to create a concentration gradient for potassium, but it's also creating membrane potential. So let's practice. For today's practice, the skill focus is argumentation. You're going to predict the causes or effects of a change in or disruptions to one or more components in a biological system based on a visual representation of a biological concept, process, or model. So let's practice. What I would like you to do now is pause your video, read the question, formulate your answer, and then push play again. Welcome back. For this question, you were looking if ADH secretion is inhibited, which of the following would initially result? And your answer should have been D. The person would produce greater amounts of dilute urine. So what should you take away from this lesson today? First, membrane proteins are required for facilitated diffusion of charge and large polar molecules through a membrane. Large quantities of water pass through aquaporons. Membranes may become polarized by the movement of ions across the membrane, along with metabolic energy, such as ATP, to help establish and maintain a concentration gradient. And finally, the sodium-potassium pump helps contribute to the maintenance of that membrane potential. I hope this video was helpful today. Until next time, thank you. Welcome to AP Daily. This is Mr. Burris, and today we're covering tonicity and osmoregulation. So what will we be learning today? For today's video, we're going to look at what are the different ways to describe relative tonicity of environments. How do differences in osmolarity impact the movement of water in a cell? How does the constant movement of molecules across a membrane maintain growth and homeostasis? And finally, what is osmoregulation and how does it contribute to the health and survival of organisms? Let's dive in. So osmosis is the diffusion of free water across a selectively permeable membrane. Large quantities of water moves across the cell membrane via aquaporons. Osmolarity is the total solute concentration in a solution. So for water, water has a high solvency ability. As review, remember, a solute is the substance being dissolved. The solvent is a substance that dissolves a solute. And a solution is a uniform mixture of one or more solutes dissolved in a solvent. So basically, a solvent plus a solute equals a solution. Tonicity. Tonicity is the measurement of the relative concentrations of solute between two solutions inside and outside of the cell. Internal cellular environments can be hypotonic, hypertonic, or isotonic to external environments. So for hypertonic, it means more solutes, less solvent, as shown in the picture. The purple circles represent dissolved molecules. Isotonic is equal concentrations of solute and solvent, as shown in the picture. Finally, hypotonic is less solute and more solvent, which you can see in the illustration. So water moves by osmosis into the area with a higher solute concentration. So water concentrations and solute concentrations are inversely related. Water would diffuse out of a hypotonic environment to a hypertonic environment, as shown in the illustration. For solutes, if they are able to diffuse across the membrane, will diffuse along their own concentration gradients from the hypertonic environment into the hypotonic environment. When a cell is in an isotonic environment, a dynamic equilibrium exists with equal amounts of water moving in and out of the cell at equal rates. So basically, no net movement of water takes place in an isotonic environment. So in the picture, is the environment hypertonic or hypotonic to the cell? Well, the answer is hypotonic, due to the fact that outside the cell, there is more water and less solute. In plant cells, osmoregulation maintains water balance and allows control of internal solute composition. Environmental hypertonicity means less cellular solute and more cellular water, which results in plasmolysis, where water leaves the plant cell. During isotonic solutions, equal solute and equal water, meaning water's equally moving in and out of the cell, results in a flaccid condition for the plant. Finally, during environmental hypotonicity, there is more cellular solute and less cellular water, so water comes rushing into the cell. This is known as turgid. The cell wall helps maintain homeostasis for the plant in environmental hypotonicity. Osmotic pressure is high outside of the plant cell due to the environmental hypotonicity. Water flows into the plant vacuoles via osmosis, causing the vacuoles to expand and press out against the cell wall. The cell wall itself expands until it begins to exert a pressure back on the cell, and this pressure is called turgor pressure. Turgidity is the optimum state for a plant cell. Now, in animal cells, osmoregulation also maintains water balance and allows control of internal solute composition. So during environmental hypertonicity, there is less cellular solute and more cellular water, just like in plant cells. However, the animal cell will shrivel. During an isotonic solution, there's equal solute and equal water. So there's equal water moving in and out in a dynamic equilibrium. And this is considered normal. This is the normal state for an animal cell. During environmental hypotonicity, there is more cellular solute and less cellular water, which is an issue to an animal cell because the animal cell could lysis, which basically means it would burst. So let's take a look at an example. A freshwater paramecium is a single-celled organism. The environment in which it lives in is hypotonic. which means that it has more cellular solute and less cellular water. Water enters the cell via osmosis. The paramecium is then in danger of the cell lysosing, so excess water collects in the contractile vacuole and then is pumped out, as shown in the illustration. So what would happen if the freshwater paramecium was placed in salt water? Take a second, think about that. What would happen? Well, water would begin to diffuse out of the paramecium because the cell is now hypotonic to the saltwater environment, and the contractile vacuole would not fill. So the cell would be at risk of shriveling. So let's practice. Pause your video, analyze the figure, read the question, and formulate your answer. When you have your answer, push play again. Welcome back. For this question, you're looking at which graph best represents the amount of starch, water, and glucose in the dialysis bag over the course of the investigation. Your answer should be A, due to the fact that glucose is able to move across the dialysis bag's membrane as well as water, but starch cannot. So what should you have taken away from this lesson? First, external environments can be hypotonic, hypertonic, or isotonic to the internal environments of the cell. Water moves by osmosis from areas of low osmolarity solute concentrations to area of high osmolarity solute concentrations. Growth and homeostasis are maintained by the constant movement of molecules across the membrane. And finally, osmoregulation maintains water balance and allows organisms to control their internal solute composition. Well, I hope this was helpful today. Until next time, thank you. Welcome to AP Daily. This is Mr. Burris, and today we're covering tonicity and osmoregulation. So what will we be learning today? For today's lesson, we're going to look at graphing. What are the components of an effective graph? What are the different types of graphs? And when should graphs be used? So let's dive in. The components of an effective graph includes a title. The title should include experiment details and what is being measured. Secondly, there should be labeled axes with units. The independent variable should be located on the x-axis, and the dependent variable should be located on the y-axis. The third component is scaling. There should be uniform intervals. The scale should be large enough to analyze data, and the scale numbers need to be located on grid lines. Fourth, there needs to be identifiable lines or bars. So either have a legend or label each line or bar. And finally, when appropriate, there should be a trend line, which is a line of best fit that shows the general pattern or overall direction of the data. So for the types of graphs, first you have a line graph, which reveals trends or progress over time for multiple groups or treatments. It also tracks changes over time, concentrations, etc. The next graph is an XY graph, also known as a scatter plot. It is used to determine relationships between two different things. You can also compare two variables that may or may not have a linear relationship. The next type of graph is a histogram. It shows how values in a data set are distributed across evenly spaced or equal intervals. You can also explore the relationships between two or more variables with a histogram. For a bar graph, simply comparing multiple groups or treatments to each other. For a box and whisker plot, you're looking at showing the variability in a sample, and it is ideal for comparing distributions in relation to the mean. The dual Y graph is used to illustrate the relationships between two dependent variables. So let's practice. During practice, you're going to need to graph. So if you have some graphing paper, that would be helpful. The skill today, we're going to be looking at representing and describing data. So we're going to construct graphs, plots, or charts. So for the first practice, what I would like you to do is pause, read the question, analyze the table, and produce a graph. Once you've produced your graph, push play, and we'll go over it. Welcome back. You should have constructed an appropriately labeled graph to represent the data in Table 1, which is the number of drops of water placed on a penny. You should have produced a bar graph because you're comparing the average number of drops of pure water to the number of drops of soapy water. Let's move to the next practice problem. Again, pause your video, read the question, analyze the table, and construct your graph. Once your graph is complete, push play, and we'll go over it. Welcome back. You were supposed to construct an appropriately labeled graph to represent the data in Table 1, which represents percent change in mass of a potato core. For this graph, you should have produced a line graph. This line graph is interesting due to the fact that you have both positive and negative numbers on the y-axis, which causes 0 to be halfway up the axis. Also, on this graph, you could determine the molarity of the potato core by the point at which it crosses 0. That represents an isotonic environment, both inside the potato and in the beaker full of sucrose and water. So what should you take away today? First, the components of a graph include a title, correctly labeled axes with units, uniform intervals, identifiable lines or bars, and trend lines. The graph types used is based on the type of data collected. Graphs can be used to show trends over time, comparisons, distributions, correlations, variability in samples, and relationships between variables. Well, I hope today's video was helpful. Until next time, thank you. Welcome to AP Daily. This is Mr. Burris, and today we're looking at tonicity and osmoregulation. So, what will we be learning today? We're going to look at how does water potential impact the movement of water? How does solute potential impact the movement of water? And finally, how do organisms maintain water balance? So let's dive in. Water potential measures the tendency of water to move by osmosis. It is calculated from pressure potential and solute potential. Water potential is represented by the Greek letter psi. And the unit of pressure is called bars. So water moves from an area of high water potential to an area of low water potential. The values of water potential can be positive, 0, or even negative. So the more negative the water potential is, the more likely water will move into the area. So if water potential is equal to -3 bars inside a cell and outside the cell, water potential is equal to -6 bars, the water is going to move from inside the cell to outside the cell due to the fact that -6 bars is more negative than -3 bars. Water potential of pure water has a value of 0 in an open container because there's no solutes, plus there's no pressure due to it being in an open container. Osmoregulation allows organisms to control their internal solute composition and water potential. So increasing the amount of solute in water will cause an increase in solute potential, but a decrease in water potential. Increasing water potential will cause an increase in pressure potential, and decreasing pressure potential will cause a decrease in water potential. So in an open system, the pressure potential is 0, so water potential is equal to the solute potential. Solute potential is equal to -ICRT. This formula is located on your formula sheet, so there's no need to memorize it. You just have to be able to use it and understand it. So for the formula, I is equal to ionization constant. So sucrose is equal to 1 and salt is equal to 2. in terms of ionization constants. C is equal to molar concentration. Molarity is the moles of solute per volume of a solution. So how much sugar is in water? R is equal to a pressure constant of 0.0831. That constant, that number, is located on the formula sheet. And finally, for temperature, temperature is in Kelvin. So you take the temperature in Celsius plus 273 and then plug it into the formula. However, an important thing to remember, the addition of solutes is equal to a more negative solute potential. So let's look at an example. A new tree is planted in a soil with a water potential of -4 bars. The roots of the tree has a water potential of -8 bars. Which way will the water move? Well, water will move from the soil into the roots since the soil has a higher water potential than the roots. So let's practice. For today's practice, we're going to be looking at statistical tests and data analysis. You're going to perform some mathematical calculations around water potential. So for this practice problem, what we would like you to do is pause your video, read the question, use the formulas, and figure out what is the water potential. When you finish, push play, and we'll discuss. Welcome back. For this question, you're looking at what is the water potential of a 0.5 molar sucrose solution at 21 degrees Celsius in an open system. For water potential, you have two equations. You have water potential equals pressure potential plus solute potential, and then you have solute potential equals negative CRT. So the first step you should have done is solve for solute potential. So for this problem, you know that 0.5 molar sucrose solution is at 21 degrees Celsius. So the ionization constant is 1 because it's sucrose. The molar concentration is 0.5. The pressure constant, R, is 0.0831. And finally, the temperature is going to be 273 plus 21. When you plug those numbers in, you should have come up with a number of negative 12.22 bars. So now we've come up with the solute potential. Now we have to look at the other part of the problem, which is pressure potential. The pressure potential, in this case, depends on the system. In this case, it's an open system. An open system equals a pressure potential of 0. With that being said, you know that the solute potential was negative 12.22 bars. So your final answer is a water potential of negative 12.22 bars. So what should you have taken away today? Well, first, water moves by osmosis from areas of high water potential to areas of low water potential. Water moves by osmosis from areas of low solute potential to areas of high solute potential. Osmoregulation maintains water balance and allows organisms to control their solute composition water potential. Well, I hope this video was helpful today. Until next time, thank you.
Good morning, good afternoon, AP Bio. This is Mr. Mansour coming to you from Tiffin Columbian High School, and this is topic 2.8, mechanisms of transport, and this is daily video 1. In this video, we will describe the process that allows ions and other molecules to move across membranes. And so what we learn, it's always important. So again, what we learn, the first thing, what is active transport? 2, why are membrane proteins necessary for active transport? And 3, how does the sodium-potassium pump and ATPase maintain membrane potential? And so when we talk about active transport is moving molecules or ions against the concentration gradient. So you may have talked about in a previous, before you got to active transport, you talked about passive transport, and that was going from high to low. In active transport, we're going from low to high, so we're going against the gradient, and that's the key. It's like we're going uphill, so you need some energy there. And so when we talk about active transport, we understand that ions, those things that have charges, they're moving from low concentration to high concentration. And so here again on my image here, high concentration, low concentration, there's that movement. That's what that arrow is representing. You're going against the gradient. If we look on this other, and when we talk about active transport, requires ATP, and that's the key. Active transport requires energy because we're going against that gradient. If we look here again, low concentration, high concentration, Again, going against that gradient, going from low to high, and again, requires ATP. And so again, you got to get that idea of active, like think of yourself moving up a hill, requiring more energy as opposed to maybe just coasting down a hill. And so when we talk about active transport, we also have to talk about metabolic energy, such as that from ATP, which we're going to talk about, and you'll talk about more in later units, is required for active transport of molecules and ions across the membrane and to establish and maintain that electrochemical gradient. Lots of words in there. If there's something you don't understand, pause the video, look it up. But again, it's required. We have to have that energy. And so membrane proteins are also necessary for active transport. So without these membrane proteins, active transport cannot occur. And so my little arrow there is pointing to a membrane protein. Those are integral proteins, which means they go through the cell membrane. You've probably already talked about this, but those are those ones that pass through. So there's our integral membrane or our membrane protein. And then the sodium-potassium pump, which is one you'll probably hear again in further part of the course, and ATP-ACE enzyme, they contribute to that maintenance of that gradient. So again, we got to focus on ATP-ACE, ATP, right? And so ATP breaks down, and as the ATP breaks down, that is what allows for this movement. So here's our ATP to ADP, right? There's that hydrolysis of ATP releasing some energy, which allows for that movement of those ions. Let's look at an example. Proton pumps. Proton pumps you're going to hear about in units 3 when we talk about mitochondria, when we talk about chloroplasts. We're also going to talk about proton pumps when we talk about somehow environment impacts phenotype. And so when we talk about, again, why this is active transport is, again, because we're going from a low concentration to a high concentration. We see those H pluses there at the bottom. There's like 2 of them. We have more H pluses up top in that extracellular fluid. And also the key is we have that integral protein, integral meaning that it's going across that cell membrane. And so we're going from low concentration to high concentration. We're trying to create this proton gradient, which is a term you're going to hear again. And again, to do all of this, we need ATP. So proton pump is one of those examples you're going to hear about quite a bit in Unit 3. And so it's essential that you learn it now. So again, that proton pump, moving those H pluses. So again, what should we take away from this brief daily? 1, metabolic energy is required for active transport of molecules and ions across the membrane. Again, that metabolic energy coming from ATP. Second, metabolic energy is required to establish and maintain that electrochemical gradient. So those gradient, again, going from low to high, that energy from ATP is used to establish and maintain it. Because if that gradient does occur, or if that gradient breaks down or doesn't, or it's not working properly, stuff will not move, and whatever process is being driven will stop. And then membrane proteins are necessary for active transport. Those integral proteins, right, those pumps, those are important for active transport to occur because those molecules have a charge. They cannot necessarily pass right through those phospholipid bilayer. They need those proteins. And last but not least, that sodium-potassium pump and ATPase contribute to the maintenance of the membrane potential. We're focusing on that sodium-potassium pump because that is one that you will definitely see in a future part of the course. And then ATPase pops up in Unit 3. So again, hopefully you found this helpful. If there's stuff in the video you're not sure of, pause, write it down, ask your teacher, things like that. So again, Mr. Mansour signing off from Tiffin-Columbian High School. Have a great day, and I'll see you in a future video.
Welcome to AP Biology, Cell Compartmentalization. My name is Paulette Unger, and I teach at Christ Church Episcopal School in Greenville, South Carolina. In this lesson, I'm going to share with you some examples of internal membranes and membrane-bound organelles and how in eukaryotic cells they compartmentalize the cell and help facilitate intracellular reactions. I'll also discuss how a change in the internal membranes or membrane-bound organelles affect the ability of eukaryotic cells to perform necessary function. All cells, whether we're talking prokaryotic cells or eukaryotic cells, have a plasma membrane that allow them to establish and maintain an internal environment that's different from their external environment. However, eukaryotic cells have additional internal membranes and membrane-bound organelles that compartmentalize the cell. And in Figure 1, several of these membrane-bound structures are labeled here for you. And if you want to learn more about the structure and function of these, you can go back and watch video lessons for 2.1 and 2.2 where I highlight the structure and function in more detail for you. But these cellular compartments allow for a variety of metabolic processes and specific enzymatic reactions to occur simultaneously, which can increase the efficiency of eukaryotic cells in carrying out their functions. So membranes minimize competing interactions. When you have several reactions occurring in the same location at the same time, molecules from these reactions can interfere with each other and maybe speed up a reaction or slow it down based on that interference. So it's valuable to have the compartmentalization that we see here. So when it comes to the case of the lysosomes contain hydrolytic enzymes that are involved in chemical digestion. But these enzymes need to function in an acidic environment, which can be nicely maintained in the membrane of the lysosome. So you can have an acidic environment inside the lysosome, while the rest of the cytoplasm can remain at a more neutral pH. And just imagine if a situation were to occur where the membranes of the lysosomes were to be damaged and the hydrolytic enzymes as well as that acidic fluid get released into the rest of the cytoplasm, and that chemical digestion is continuing to occur. This could lead to some severe cell damage and possibly cell death. Membrane folding also can be maintained in these different membrane enclosed organelles and maximize the surface area for metabolic reactions to occur. So we can see how that inner membrane in figure 1 of the mitochondria is highly folded, and this will increase the surface area for electron transport and ATP synthesis. And so that folding will maximize the amount of ATP that the mitochondria can generate for the cell to do work. And if you reduce that folding in any way, or if it's damaged to that folding, it's going to be less efficient at making energy, and that can ultimately impact the entire cell. We see the same membrane folding occurring in the chloroplast that is found in plant cells and other photosynthetic algae. And so the thylakoids that you see here in figure 2 in the chloroplast model are highly folded membrane compartments that increase the efficiency of the light-dependent reactions. So you have these photosynthetic pigments and these chlorophyll pigments in the membranes of the thylakoid capturing light energy for those light-dependent reactions to occur most efficiently. So again, if there was any loss in the membrane structure there and that increased folding, it would just reduce the amount of light energy captured by the plant cells. So it's important for scientists to construct arguments to explain principles or concepts or ideas and reason through that. And part of argumentation is making predictions or expected results based on a disruption or change to a system. So here's an AP practice question. I'm going to ask you to pause the video, and when you have your best answer, press play and we'll discuss it. All right, so let's see what we have. So some viral infections can lead to the rupture of lysosomal membranes. Which prediction of the effect of this disruption of cellular compartmentalization is most likely correct? Choice C, hydrolytic enzymes will be released, which will cause cell death. So some key takeaways here are eukaryotic cells contain a variety of membrane-bound organelles that compartmentalize intracellular processes and enzymatic reactions and ultimately increase the efficiency of eukaryotic cell function. Internal membranes facilitate these cellular processes by minimizing competing interactions or by increasing surface area through membrane folding. And any loss of these intracellular compartments or changes to these membranes can hinder proper cell function. Thank you, and I hope this was helpful, and if you need more time, just rewind. Welcome to AP Biology, Origins of Cell Compartmentalization. My name is Paula Dunger, and I teach at Christ Church Episcopal School in Greenville, South Carolina. In this lesson, I will discuss some similarities and differences in compartmentalization between prokaryotic and eukaryotic cells, and then talk about how the mitochondria and chloroplast evolved from previously free-living prokaryotic cells via endosymbiosis, and share with you some evidence to support this claim. There are two main cell types that make up living organisms, prokaryotic and eukaryotic cells. Both of these cell types have plasma membranes as shown here. So you can see the plasma membrane circled in the prokaryotic cell in figure 1 and the plasma membrane of the eukaryotic cell in figure 2. And the plasma membrane functions to separate the internal environment from the surrounding environment. Now, the prokaryotic cells lack internal membrane-bound organelles, and so the only membrane that the prokaryotic cell has is that outer plasma membrane. However, prokaryotic cells do have internal regions, such as the nucleoid region shown here, that contains its genetic material. Eukaryotic cells, on the other hand, do have more extensive internal membranes and membrane-bound organelles that compartmentalize the cell. And the genetic material is contained within a membrane-bound nucleus. And this is a distinguishing feature of eukaryotic cells from prokaryotic cells. So how did these membrane-bound organelles that we see in eukaryotic cells evolve? Well, the nucleus and other internal membranes like the ER are theorized to have formed from the infoldings of the plasma membrane. So you can see those structures on this initial cell. Mitochondria and chloroplasts, however, evolved from previously free-living prokaryotic cells via endosymbiosis. So I'm going to use the mitochondria here and its evolution to talk you through what endosymbiosis is. So a free-living aerobic prokaryote was engulfed by an anaerobic cell through endocytosis, and that's shown here in this diagram. So we have this aerobic proteobacterium that was taken in by the anaerobic eukaryotic cell through endocytosis. Then the engulfed prokaryotic cell, however, did not get digested by the engulfing cell. So it was actually more of a win-win situation. So the aerobic bacteria that was ingested took care of oxygen that was available, and the anaerobic cell could have provided food molecules, and this would lead to energy and metabolic reactions that would have benefited this cell. And then over time, the engulfed cell lost some of its independent functionality, however, and became the mitochondria for that eukaryotic cell. So you can see that labeled there. And it was a similar evolution for the chloroplast, except in this case, a free-living photosynthetic prokaryote was engulfed by another cell through endocytosis. And again, it was another mutually beneficial relationship here where the photosynthetic bacterium that was engulfed would provide energy for the cell, and the cell could provide protection for that ingested photosynthetic bacterium. But again, over many generations, the engulfed cell lost some of its independent functionality and became the chloroplast of eukaryotic cells. So what is some evidence that supports the evolution of mitochondria and chloroplasts by endosymbiosis? Well, let's take a look at the relationship between the functions of endosymbiotic organelles and their ancestors. So we have the mitochondria and chloroplasts here in these figures, and both mitochondria and chloroplasts have double membranes. So that's the first piece of evidence. These double membranes function to regulate the passage of materials into and out of the cell and help maintain a stable internal environment. So you can see here in these figures, they have these two double membranes showing how they were once free-living and then engulfed through endocytosis, which would lead to that double membrane structure. And like prokaryotic cells, mitochondria and chloroplasts both have their own circular DNA, which encodes genetic information, and they can reproduce by similar processes. And so you can see here that circular DNA in both of those structures. And also, both mitochondria and chloroplasts contain their own ribosomes, as shown here, which make proteins for the mitochondria and chloroplasts, similar to how prokaryotes have ribosomes to make proteins for them. So now let's practice the skill of argumentation. And in this part of argumentation, after a claim is made, it's important to support a claim with evidence from biological principles, concepts, processes, and data. So here's an AP multiple choice question for you to practice the skill of supporting a claim. So hit pause, and when you have an answer, press play again, and we'll see what you got. Okay, so the question's asking the mitochondria are found in most eukaryotic cells and contain their own DNA and ribosomes that are similar to those typical of many prokaryotic cells. Which of the following statements is justified by these observations? And the correct choice is B. An ancestral cell most likely engulfed anaerobic prokaryotes in a relationship that proved beneficial to both cells. So there's that mutual symbiotic relationship that came about. Some key takeaways here are that both prokaryotic and eukaryotic cells have external plasma membranes. However, whereas the prokaryotes only have internal regions, where specialized structures and functions can occur, such as the nucleoid region containing the DNA that I showed you previously, eukaryotic cells have additional internal membrane-bound organelles to compartmentalize the cell and help with cell efficiency. According to the theory of endosymbiosis, a previously free-living prokaryote was engulfed by another cell through endocytosis, and after living together symbiotically for some time, the once free-living prokaryote lost its independent functionality and gave rise to either the mitochondria or the chloroplast. And there's several pieces of evidence supporting this claim, such as the presence of double membranes in both mitochondria and chloroplast, circular DNA in both those cell parts, and ribosomes as well in both the mitochondria and the chloroplast. So thank you for watching, and I hope this video is helpful.